Welcome back. We will continue with the life cycle of Taenia solium. We saw that the proglottids, the gravid proglottids, it comes out of the human intestine through the feces and it gets desiccated, it's broken down, releasing the oncospheres into the surrounding medium, right? And these oncospheres ingest the secondary host, that is a pig. This is what we have dealt in the previous video. And usually pig forms a secondary host, but humans may serve as a secondary host in some cases when they ingest the oncospheres along with inadequately cooked food or maybe along with raw vegetables. And even O2 infection is also found in Taenia solium in certain cases where a person already serving as a primary host may show this auto-infection. Here what happens is, due to reverse peristalsis, the proglottids which are detached from, here you can see the detached proglottids, isn't it? The proglottids which are detached from the adult tapeworm, they may be carried back to the stomach because of reverse peristalsis. And these, what you call proglottids, once it reaches the stomach, it will release the oncospheres. And this causes an auto-infection in the case of humans. Okay. Now, what happens after the oncospheres enter the secondary house? This is what we are discussing, isn't it? On reaching the stomach of the secondary house, the oncosphere, they lose their embryophore and the basement membrane. I'll show you the, yeah. So this is the oncosphere, isn't it? So once this oncosphere reaches the stomach of the pigs, it loses the embryophore, the outer covering over here, and the shell and all, and in the stomach, what happens is because of the action of the acidic juices, these membranes gets broken down, it gets completely digested. Now, what happens? The hexacanth inside passes into the small intestine. The hexacanth, it comes out. So that because of the losing of these membranes, the hexacanth is liberated, and these hexacanth, it moves into the small intestine where the two persisting hexacanth membranes are also lost by the action of alkaline juices in the intestine. Now, the hexacanth is activated by the presence of bile salts, and they, by the activation, they bore into the intestinal epithelium. Okay, they bore its way through the intestinal epithelium and reach the submucosal blood vessels. And this is made possible by the these hooks on the hexacanth. Okay, the hooks present on the hexacanth helps in penetrating through the intestinal epithelium, reaching the submucosal blood vessels, and these happens because of the hooks mainly. And along with that, there is another structure which helps in this penetration, and it is the penetration glands. Okay, the hooks helps in anchoring the hexacanth to the intestinal wall, while the secretions of the penetration gland, it helps in dissolving the intestinal tissues and helping them for easy penetration. Okay, so the whole entire process of oncosphere reaching the intestinal region and then penetration of these hexacanth into the intestinal wall, all these happens maybe like by within 10 minutes, and the whole process gets completed in about 10 minutes. Okay, and once this whole process is completed, they don't require hooks or such kinds of structures anymore, so that is being shed off. Once the what you call hexacanth, it reaches the blood vessels, they don't need the hooks anymore. So what happens is, it is actually completely shed off. Now they are devoid of any hooks on the hexacanth. I hope that part is clear. Okay. Now the submucosal blood vessels carry these hexacanth to the liver via the hepatic portal vein. So the blood which reaches the liver through the hepatic portal vein may also carry these hexacanth, and from there it may reach the heart and enters the arterial circulation. Finally, it may reach the muscles, the striated muscles, usually of the tongue, the shoulder, the neck, the thigh, etc., where it gets settled to develop into a bladder worm. Okay, this is known as a bladder worm, and it later developed into the cysticercus state. Okay, now that is where we are going to see the next phase, that is the cysticercus or the bladder worm formation, how it is formed from the hexacanth. Now the hexaconch, which is now devoid of any hooks, they absorb nutrition from the horse tissue. Which is the horse now? The host is the secondary host, the pig. Okay, so it actually absorbs the nutrients from the host tissue. They grow in size, and later what happens, what we can see is a central cavity, an invagination appears in the hexaconch as the cells in that region break down. You can see here as an invagination is developing. Okay, this is a central cavity is developed. This cavity enlarges and becomes filled with a fluid consisting mainly of the blood plasma of the host body, okay, of the host. And this fluid filled vesicle, it is otherwise referred as a bladder. Okay, this kind of a structure is what is referred as a bladder. Okay, this you can see, right? This is known as a bladder. Fine. And this bladder has a thin wall which consists of an outer layer of thick syncytial protoplasmic mass, or what we can refer as a cuticle, and the inner mesenchymal layer. Okay, so at this point we can see the anterior end, the wall thickens and invaginates. Okay, and the invagination, which looks like a hollow knob, this one. Okay, here you can see this invagination, and this slowly it will get transformed into an inverted scolex. The scolex of the adults, you remember the structure. Okay, almost this slowly gets differentiated into the scolex region, and this part is invaginated, or what you call inverted scolex, and this scolex is referred as a proscolex, inverted scolex, which is formed from the invagination of the bladder stage, okay, bladderworm stage. So these inverted scolex or the proscolex may possess the, you can see here the hooks are there, the suckers are there, the rostrum is there, the suckers are here, isn't it? So all these structures are present, and it is referred as a proscolex. And this embryo at this stage with an inverted proscolex, it is referred as a bladder worm. I hope that part is clear. Okay, this is known as a bladderworm stage. Okay, here what happens is the bladderworm is of cysticercus type, which is characterized by a large vesicle and one scolex. Okay, so you can see this is of a cysticercus type. Fine. Now the cysticercus develops in the adult tapeworm only when ingested by the human host. Okay, in this cysticercus is actually you can see this is, it is formed from this one. Okay, it is a cysticercus type. Now this cysticercus stage, it will develop into the complete adult tapeworm only when it reaches the, so it is a cysticercus type. Where it is present in the striated muscles, right? And it will remain as such in the cysticercus stage itself till it enters the human intestine, right? So in the pig's body, these cysticerci in plural, it may lead an inactive life, but remain viable for several years. That is, for several years it can remain in inactive life, and then whenever a favorable time reaches, that is, whenever it can reach the human intestine, it will develop into the adult tapeworm. This is what happens. Okay, and the pork or the pig's flesh, which contains the cysticerci, they are known as measly pork, right? And the name has come because of the spotted appearance, because of the presence of these cysticerci in the muscles. You can see spotty appearance it gives, and this, because of that, it is known as a measly pork, right? Now the human host gets the infection by eating these undercooked measly pork. The cysticercus, it becomes active on reaching the human intestine, as already mentioned. Okay, when it reaches the small intestine, it becomes active. Now, till then, what stage it was in? It was in a what they call proscolex stage. So the proscolex, it everts or it evaginates. It evaginates, like whatever was inside the proscolex, it came out. That is how the structure is formed, isn't it? So it everts. You can see here the evaginated scolex, and it shows off or exhibits the hooks and the suckers and all, and with that they anchor onto the intestinal wall. Okay, now the neck now begin to proliferate, that is, it starts to bud off new what we call segments, proglottids, and then the sooner or later the bladder, this is the bladder, okay, you can see here, this is the bladder, right? And this is the evaginated scolex of the, you You can see this is the evaginated scolex and this is the bladder. But as soon as it attaches onto the intestinal wall, the neck starts to bud off new segments, right? New proglottids are formed. And then sooner or later, what happens is this bladder is completely detached and it is completely get digested off. Okay, so then slowly it increases in size with the addition of new proglottids and it gets completely grown into an adult tapeworm. This is what happens in the life cycle. So we can see that the adult tapeworm, it is self-fertilization, zygote is formed, the zygote develops into a morula-like stage in the uterus, right? And from there, it develops these what we call morula-like stage develops like hexacanth condition, that is with three pairs of hook-like structures on the multicellular structure. And this hexacanth, it gets surrounded by membranes like the embryophore and the hexacanthal membrane, and it transforms into what is known as an oncosphere. It is in this oncosphere condition, it gets laid inside the uterus of the proglottid, and those proglottids with these oncospheres, they are known as ripe or gravid proglottid. These ripe proglottids are being released along with the fecal matter into the surrounding medium, into the surrounding, right? And from there, it gets entry into the secondary host when these pigs feed upon the night soil of humans. Okay, so these what you call oncospheres get inside the reach the intestine. Okay, and you can see the encysted hexacanth. Reaching over there in the intestine, the proteolysis will break down the membranes of the oncosphere and it releases the hexacanth. You can see, I'm sorry, yeah, the hexacanth. Okay, you can see the hexacanth with all those hooks here, right? And then these hexacanth, it will undergo the transformation into the bladder worm, where the invagination it will develop into the proscolex, and the whole stage is known as the Bladderworm stage. And from there, this bladder worms, it will migrate and get deposited in the skeletal muscles or the striated muscles in the and this such kind of muscles of the pigs, if it, when it is eaten by humans, these what we call the proscolex stage or the bladder worm will get entry into the human intestine. Reaching over there, these bladder worms, the evagination of the scolex happens and the scolex is visible now, and this will attach onto the wall of the intestine with the help of the hooks on the what you call rostrum, and with the help of the scolex, and with the help of it will remain attached to it. And sooner what happens is the bladder is detached from the part, and new segments get added by budding of the neck. So this is how the whole life cycle is completed. Now, what are the parasitic adaptations shown by Taenia solium? As we have already seen in the case of fasciola, that the parasitic adaptations can be considered under two headings, that is degenerations and neoformations. Here also we can see under degenerations there is loss of locomotor, digestive, circulatory, respiratory organs, because it doesn't require any such structures because they are inside the body. The complete life it is spent inside the host body, so they don't need any kind of structures like this. Similarly, absence of brain and receptor organs, they don't need any what you call receptor organs for perceiving the change in the stimuli, because they are always in the same kind of a situation, right? Then neoformations, presence of rostrum, hooks and suckers for adhering onto the what you call this mucosal layer of the intestine, resistant tegment for protection. Then anaerobic respiration, because in the intestine free oxygen is not available, it is in an anaerobic condition, so they have developed anaerobic mode of what you call respiration. Because of the flattened body, they have a large surface area, and this large surface area, the rest of the process, the absorption, etc., is being done. Okay, and then hermaphroditism. Obviously, this is self-fertilization, and it is very difficult to find another individual of the same species for reproduction. So hermaphroditism helps in increasing the chance of reproduction. Okay, so these are the parasitic adaptations. Then pathogenesis, when we see teniasis and cysticercosis are the two conditions which can lead, which can be resulted by the infection of Taenia solium in human beings. Okay, so this is all about Taenia solium. Thank you.